afternoon. Joe, you've got a good crowd here. I'm very happy about that. Well, I'm very excited to, and actually it's really, what should I say, rewarding to welcome Joe Mancius to give a talk here. Joe came to our division in 2016. He started in 2016, so it's been a few years. But let me tell you before, so Joe is a radiation oncologist, a bona fide radiation oncologist who actually sees patients and also publishes cell science Nature papers. That's a pretty rare combination. So he did his PhD in Weill Cornell, and he did it on structural work with Jonathan Goldberg. Then he came to HMS, did his residency, and was a postdoc with Wade Harper and Alec Kimmelman, who was part of radiation oncology here in those days. And Joe published this seminal Nature paper. I believe he's going to talk a little bit about that, so I won't take it more. And he's been a fantastic member of our community and has been productive in many fronts. He's received many awards. I'm not going to go over all of them, but the more prestigious ones are the Damon Runyon Innovation Award, and he's the Sidney Kimmel Scholar Award are the ones which come to mind. But it's the science that he does which is spectacular, and I'll give it to him.
Thank you, Dipanjan, for the introduction as well as the invitation to present. I'm excited to be here and present in front of my colleagues, both from the clinic as well as from the laboratory. Before getting started, I'll just have my disclosures here. One of the difficulties of diagnosis of pancreatic cancer is the intense therapeutic resistance of this disease to all therapies that we have, whether it be chemotherapy, radiation, targeted therapy, or immune therapy. And much work has gone on to understand this resistance, and much of it relates to the tumor cell's altered cell survival and altered metabolic pathways that promote its proliferation and survival. And one of those pathways is an extra reliance on autophagy. So autophagy is a conserved catabolic cellular process that serves to degrade cells' own proteins and organelles via the lysosome. So in a simplified schematic, a phagophore, a double membrane structure, subsumes within it cargo, fuses on itself to form an autophagosome, which then fuses with a lysosome that delivers its degradative enzymes. The contents then are degraded in this autolysosome and can be recycled and used by the cell as energetic intermediates or building blocks. And so this process is going on at some basal level in all cells, but can be further stimulated under periods of stress, such as nutrient deprivation. Derangements in this pathway have been linked to neurodegeneration as well as in cancer, with the poster child really being pancreatic cancer. Now, if we're thinking about targeting this pathway, unfortunately, all that's available right now, FDA approved wise, is hydroxychloroquine, which is a weak inhibitor of lysosomal acidification. So really, work in our lab has centered around trying to understand what the role of autophagy is in pancreatic cancer, with a focus on what's being degraded via autophagy, with an eye towards more targeted autophagy inhibition specific to pancreatic cancer. Other work in the lab is trying to define and target resistance to various new therapies, most notably with the development of new KRAS inhibitors that are coming to the clinic now for pancreatic cancer. There's going to be some effect of those, but unfortunately, monotherapy is not going to be enough. There's resistance that's going to be developed, and we want to get ahead of that and understand, as many other labs do, what are effective combination therapies. A lot of our work revolves around using mouse models of pancreatic cancer, whether those be genetically engineered or transplantable models, and an extra reliance on using mass spectrometry-based quantitative proteomics to answer some of our questions. We're fortunate to be members of the Hale Family Center Pancreatic Cancer Research and collaborate extensively within this center. So the seminal work defining the importance of autophagy in pancreatic cancer was completed by the lab of my postdoc mentor, Alec Kimmelman, as well as several other labs, including Eileen White and Nabil Bardisi. And the first observation is there was an elevated level of autophagic flux in pancreatic cancer cell lines. So here, basically looking at one of the proteins that mark the surface of the autophagosome, ATG8 family member proteins, you can see that there's an increase in the amount of autophagic puncta in pancreatic cancer cell lines when compared to a normal pancreatic ductal epithelial cell line. And similarly, when you look at tumors from human specimens in the adenocarcinoma, you can see an elevated level of this protein and an increase at the pre-invasive stages. So it suggests a higher level of autophagy, but what's the function of this? What's the functional relevance? So using hydroxychloroquine or chloroquine in a pancreatic xenograft model, they demonstrated that there's an improvement in survival and a delayed tumor progression. And then targeting one of the core autophagy genes, ATG5, in the context of a genetically engineered mouse model, again showed an improvement in pancreatic cancer-specific survival. So this all sort of was the foundational work showing that autophagy is elevated basally in pancreatic cancers and is required for tumor growth. And indeed, this has then been labeled as one of the hallmarks of pancreatic cancer therapies targeting this nutrient scavenging pathway. Unfortunately, with only hydroxychloroquine available as a therapy, the results in the clinic have been less than promising. One of the first phase 2 trials evaluating hydroxychloroquine in patients with advanced metastatic disease was run here, led by Brian Wolpin and Alec Kimmelman. And unfortunately, the hydroxychloroquine monotherapy itself was inconsistent in terms of its autophagy inhibition levels and demonstrated negligible therapeutic efficacy. Further phase 2 clinical trials, including in pancreatic cancer and combinations with chemotherapy, as well as in other autophagy-dependent cancers, similarly show that hydroxychloroquine really isn't the answer if we want to target autophagy, given its ineffective and unclear PK and PD properties. So we really need new autophagy inhibitors and need to think about what are going to be the right combinations of autophagy inhibition. To that end, there's been some new excitement in the field over the last few years. In particular, given that pancreatic cancers are driven mostly by oncogenic KRAS, there was an interest in understanding what happens to autophagy when you inhibit the KRAS MAP kinase pathway. And what was interesting is that even though pancreatic cancers have a high basal level of autophagy, when you inhibit KRAS here by knocking down KRAS or by inhibiting KRAS G12C with new pharmacologic inhibitor strategies, you can see that there was a further increase in autophagic flux. This work was completed by Channing Durr's lab, and in a companion study by Martin McMahon's lab, they identified a similar mechanism and actually evaluated the combination of a MEK inhibitor, trametinib, with hydroxychloroquine in a N-of-1 study. It showed a dramatic response in tumor marker as well as a radiographic response, suggesting that there was good synergy between targeting the KRAS MAP kinase pathway and autophagy. And so this led to a trial, again, run here by Brian Wolpin, evaluating ERK inhibition alone or in combination with hydroxychloroquine. So this is completed, and we await the results, but really, I think we're all looking forward towards these new KRAS inhibitors and the next line of autophagy inhibitors that might be more effective. Another exciting discovery was that pancreatic cancers actually degrade MHC1 via autophagy. And so by decreasing the level of MHC1 on the tumor cell surface, this is an immune evasion mechanism and does contribute to the refractoriness to immune checkpoint blockade. And so we think about inhibiting autophagy and stabilizing MHC1 complexes on the cell surface. This may lead to increased anti-tumor T-cell immunity and improved response to immune checkpoint blockade. And that's the subject of ongoing clinical trials. This lab work was led by Alec Kimmelman and Rishika Pereira, with our lab collaborating. When I entered the field during my postdoctoral fellowship, we were still thinking about what are going to be the new targets for autophagy inhibition beyond hydroxychloroquine. We decided to focus in on what is being degraded via autophagy in pancreatic cancer. It had previously been thought that autophagy was a bulk, non-selective degradation mechanism whereby whatever was near this forming phagophore was going to be degraded. But there was a growing realization at the time that there's selectivity built into this pathway, mainly in the form of so-called selective autophagy receptors or adapters that can selectively identify proteins that are bound for autophagic degradation. But the full complement we didn't think was discovered. And we thought that it might be that pancreatic cancers are using autophagy to degrade specific proteins or even organelles that might promote their proliferation and survival. So to answer these questions, we took a quantitative autophagosomal proteomics approach, whereby we enriched autophagosomes from pancreatic cancer cell lines using a differential gradient centrifugation technique. We validated this using electron microscopy, showing these double membrane autophagosomes and more electron-dense autolysosomes. We used quantitative proteomics in a multi-step filtering process in order to identify what were the most highly enriched proteins in these autophagosomes. And we're happy to identify that many of the known autophagy proteins were in these autophagosomes, including these ATG8 proteins that stud the outside and inside of autophagosomes, as well as cargo that are within the autophagosome, the known autophagy receptors at the time, as well as some of the autophagic machinery. In addition to these known proteins, there were a number of candidates that had not been previously associated with autophagy, in which we wanted to understand what their potential role was in autophagy. One of them that stood out to us was called NCOA4, in part because it had previously been characterized as a nuclear receptor coactivator, so left us scratching our heads a bit as to what it could be doing in autophagosomes. So to figure that out in an unbiased way, we used interaction proteomics to figure out what NCOA4 was binding. And by pulling down NCOA4, we identified ferritin heavy chain and light chain as binding partners of NCOA4, as well as HERC2 and neural4, which are part of an E3 ligase complex. And we look back in our autophagosome proteomics data sets, not only do we find obviously NCOA4, but we also found ferritin heavy chain and light chain enriched in autophagosomes, but did not find HERC2 and neural4. Validated these results by Western blot and confirmed that the NCOA4 was colocalized with ferritin in ATG8 structures that are representative of autophagosomes. So this led us to think a little bit, well, what is ferritin? For a long time, it's been known that ferritin is the intracellular iron storage complex. And the reason this is important is iron is critical for all forms of life, especially mammalian life. Iron is critical for many different processes, including RNA, DNA metabolism, mitochondrial metabolism. And so you need iron. But there's a flip side to it. Too much iron is going to be toxic because it can participate in the Fenton reaction and generate oxidative stress in the cell. So the cell's solution to this is creating this spherical complex of ferritin, heavy chain, and light chain subunits that has a hollow inner core in which you can store iron in an inactive form. But what hadn't been known is how you get the iron out when you need it. There were some early clues in that ferritin and iron were found in lysosomes and electron micrographs from early studies. And in a study around the time when we were studying this, there's a suggestion that autophagy might be important for getting ferritin to the lysosomes. Here, they were basically evaluating what happens when you starve cells of iron with an iron chelator, DFO in this instance. And you can see that ferritin decreases over time. But when you knock out or knock down ATG5, one of the critical machineries of autophagy, you can see that there's a rescue of ferritin turnover, so suggesting that ferritin is brought to the lysosome by autophagy. So our hypothesis, given that NCOA4 and ferritin were interacting, was that NCOA4 could be the selective autophagy adapter for bringing ferritin to the autolysosome, where it could be degraded, iron released, and used by the cell. So to evaluate that, we used a similar setup where we loaded cells with iron, ferric ammonium citrate. This induces the translation of ferritin, and then we starved the cell of iron using multiple different iron chelators, DFO, BPS, and DFP. And you can see in the control condition, there's a decrease in the ferritin levels so that the iron is released from the ferritin and can be used by the cell. In the NCOA4 knockdown conditions, though, we saw that there was a rescue of this ferritin turnover, suggesting NCOA4 is important for the turnover of ferritin. We performed the same experiment and then looked at ferritin localization, but added lysosomal protease inhibitors to prevent the degradation of lysosomal contents. And so here in the control condition, you're able to see that ferritin accumulates in lysosomes marked by LAMP2, but in the NCOA4 knockdown conditions, you no longer have as much colocalization of ferritin in the lysosome. So this suggests that NCOA4 is critical for the turnover of ferritin and the localization of ferritin in the lysosome, so thereby nominating this as the new selective autophagy adapter for ferritin. So what are the consequences of not having NCOA4? So as I mentioned, it's critical that you have iron for many different iron processes, and there's this so-called labile iron pool that is critical for being shuttled to various different locations in the cell to be incorporated into iron sulfur cluster proteins. And so there are intricate sensing mechanisms that detect the level of iron that's free in the cell. One of them is this iron response element binding protein, IRP2. For the purposes of this experiment, it's important to know that under low iron conditions, IRP2 is stabilized, whereas in high conditions, it's degraded via FBXL5. The reason this is important is we'll look at the IRP2 levels as a marker of the iron levels in the cell that are free in the context of N2A4 wild type or knockdown conditions. The second thing is that IRP2 is important for stabilizing or inhibiting the translation of transcripts. For the purposes of this experiment, understanding that IRP2 binds to the 3' UTR of transferrin receptor, thereby stabilizing its transcript to allow for increased translation of transferrin receptor, which is a receptor on the cell surface that brings iron from outside the cell, which is what you'd want to do if you had a lower level of free iron. So we were excited to see that when you knock down N2A4, you increase the level of IRP2 in multiple different cell lines. So this suggests there's a lower level of free iron in the cell. And then there was a compensatory increase in the transferrin receptor to get more iron in from outside the cell. Another consequence that we thought of with regards to loss of N2A4 is if there's a lower level of iron in the cell, then there might be less sensitivity to oxidative stress-induced cell death. And so at the time, what we evaluated was stressing the cells with hydrogen peroxide in an N2A4 knockdown condition. And we saw, as we would have expected, with less iron available, that there was a decreased sensitivity to this hydrogen peroxide-induced cell death. So at this point, we were excited that we'd identified a new selective autophagy process, and we were also excited to see that another group shortly after ours, led by Lone Murphy, then at Novartis at the time, discovered the same process. From there, we were interested in sort of completing and understand the regulatory mechanisms of N2A4, and work I'm not going to show. It's really the levels of N2A4 that dictate flux through this pathway. And the levels are actually in part modulated by that HERK2E3 ligase that I'd mentioned as a binding partner of N2A4. And we discovered that actually N2A4 binds to iron. And so when the iron levels are high in the cell, HERK2 recognizes N2A4 to degrade it via the ubiquitin proteasome system. This allows for the cell to prioritize putting the iron in ferritin as opposed to turning it over via ferritinophagy. So in the ensuing years, there's other groups that have been interested in the biochemistry and the cell biology of N2A4, and there's been some additional wrinkles, including that N2A4 and ferritin enter this phase-separated situation, which is what is actually autophagocytosed via classical autophagy, but there's also this endosomal microferritinophagy pathway that's been described as well. In addition, our finding that N2A4 binds to iron was validated, but it was actually that N2A4 incorporates an iron-sulfur cluster, and that is what is recognized by HERK2. And by virtue of having it being an iron-sulfur cluster, not only do N2A levels are sensitive to the iron levels in the cell, but they're also sensitive to the oxygen levels, where in a low oxygen level, there's going to be degradation of N2A4, and at a high oxygen level, there's stabilization of N2A4. To the point I was beginning my lab, we had a number of unanswered questions with regards to N2A4, including what were the molecular determinants of binding and modulation by post-translational modifications of the N2A4 ferritin interaction, as well as HERK2, how N2A4 and ferritin are trafficked around the cell, whether there's any transcriptional regulation. And really what we wanted to move to, though, was we thought that the intracellular regulation of iron homeostasis is critical, but our real questions when we started this project were, can we figure out anything that's important for pancreatic cancer? So we wanted to move towards the physiological function of N2A4 and systemic iron homeostasis and erythropoiesis, as well as trying to understand if there was any role of N2A4 in pathologic conditions, such as hemochromatosis, which is a condition of iron overload, or pancreatic cancer, which our lab is primarily focused on. At the time, there was also this discovery of an iron-dependent form of cell death, ferroptosis, which others have shown, and we have shown as well, that N2A4 can modulate sensitivity to ferroptosis, and so there's ongoing projects in our lab evaluating that. But I'll tell you today about several different stories. First, our use of conditional knockout mouse models to understand the physiologic role of N2A4, a new discovery of the role of N2A4 in the cell-autonomous function of autophagy in pancreatic cancer, and in new work in collaboration with the Frank Lab, the molecular structure of N2A4 and ferritin and how that interface developed. So why would we want to study the role of N2A4 in physiology? So as I mentioned, iron is critical for basically all processes in life. And so there's tight regulation of iron metabolism, not only on the intracellular level, but also systemically. So iron enters in via the gut, is stored in the liver, can utilized in the bone marrow for production of red blood cells, recycled by macrophages, and all of this is controlled by hormones produced in the kidney and the liver. And so, there's this intricate network, and we wanted to understand how N2A4 fits into this. Now, a couple other groups had actually developed constitutive systemic N2A4 loss models and shown, as expected from our cell culture experiments, that loss of N2A4 leads to ferritin accumulation and that there was a mild anemia. But what you can't answer with a constitutive systemic N2A4 loss is what is the role of N2A4 basally in an adult? Is this important to have basal ferritinophagic flux ongoing? And what is the erythroid autonomous role of N2A4? Is there any importance of this in the red blood cell compartment? So to answer those questions, we developed a conditional knockout mouse model and crossed that with a tamoxifen-inducible UBC-CRE2 model that allows temporal control as well as systemic ablation of N2A4, as well as a erythroid-specific EPOR-CRE allele. With the tamoxifen-inducible model, we're able to see nice loss of N2A4 in various tissues, and we set up an experiment to look at what are the acute effects of loss of N2A4 as well as at a later time point to understand whether or not there's flux through this pathway basally. And indeed, when we looked at that early time point, the first thing that we saw in the liver is that there was an accumulation of total iron. So this is not just free iron, this is iron that's trapped in ferritin as well. And with that, there was actually a concomitant decrease in the serum iron. So this is the iron that's available for use by the animal. And presumably, because you've ablated N2A4 and reduced the flux of ferritin through the lysosome, you're no longer getting the iron out of the liver ferritin, and so you decrease acutely the level of iron that's available systemically. And we also saw, as expected, there was an accumulation of ferritin in the liver, but when we looked into other organs, we didn't see as much accumulation of ferritin, so suggesting that there's variable flux depending upon the tissue. When we looked later on, both by immunoblot and immunohistochemistry, we saw accumulation not only in the liver of ferritin, but also in the pancreas, as well as many other organs. So again, suggesting that there's variable flux in different tissues and something that we don't really understand as to how this is actually programmed in various tissues. What we then looked at are the erythroid parameters to understand what the effect of systemic acute ablation is. And we saw at the early time point that there was a general mild anemia with decreases in red blood cell numbers, decrease in hematocrit, hemoglobin. But as you looked later on, the mice actually compensated. And so there was no longer anemia multiple weeks on. And we're not showing this, but we determined that there was a compensatory increase in the HIF2 alpha erythropoietin axis in order to compensate for loss of N2A4. What this didn't answer whether or not this was going on in the erythroid compartment or it was a non-autonomous function of N2A4 in terms of supporting erythropoiesis. And so we paired the N2A4 conditional allele with an EPOR-CRE allele to lose N2A4 in the erythroid compartment and looked at 2 time points. First, the postnatal day 10, when mice are still accumulating their iron stores and where getting the iron out of ferritin might be still critical. And indeed, we saw a more profound anemia than when compared to an adult mouse, where we actually saw a very mild microcytic anemia that was compensated again by elevation of the HIF2 alpha erythropoietin axis. So, from this initial study, we basically concluded that N2A4 flux is active systemically at sort of basal conditions and important for maintaining systemic iron homeostasis. But as it relates to erythropoiesis, it's only a minor role in terms of the cell autonomous function, and that there are multiple, as one would expect, compensatory mechanisms in order to drive red blood cell production. Really, what we're here for today is to talk about cancer, and wanted to get back to this as we were starting my lab, and really understand, did what we discover in pancreatic cancer cell line autophagosomes, is that relevant to pancreatic cancer pathogenesis? So we hypothesized that N2A4 is promoting iron bioavailability, and that fuels tumor progression. And the reason why we thought that is that there's been a history of studying iron addiction in cancer. Multiple cancers have been shown to be addicted to higher levels of free iron. Those included ovarian, breast, colon, and pancreatic. And there's an iron signature that's been associated with poor survival in multiple cancers. So what are the mechanisms of iron addiction? How do you get more iron in a cancer cell? So it's using the same mechanisms that are normally active. That could be an increase in import of iron via transferrin receptor, a decrease in the export via ferroportin, or, interestingly, a decrease in the storage of iron and ferritin. In terms of non-autonomous mechanisms, there actually can be feeding of iron from non-tumor cells, such as tumor-associated macrophages or fibroblasts in the microenvironment. That might be another mechanism of iron addiction in cancer. As to what cancers are utilizing the iron for, it's the same things that you would use it for in a normal cell. That would be for proliferation, thinking about RNA, DNA metabolism via ribonucleotide reductase. Multiple aspects of mitochondrial metabolism rely upon iron, mainly in the form of incorporation of iron into iron-sulfur cluster-containing proteins. In addition, iron-sulfur clusters are critical for DNA damage, many DNA damage, repair mechanisms, so maintaining genomic integrity. So there's been a thought for a long time is if you can target iron in cancer, you might be able to affect cancer growth. There's another side to this, though. When cancers sort of increase their reliance on iron, they also create a vulnerability. And so people have been interested in understanding whether these higher iron levels that are in cancer cells might actually lead to a liability of this ferroptosis, this iron-dependent form of cell death that many people are studying. So with this as the backdrop, we also saw recently that looking at this iron mRNA signature and TCGA data at another group, Eric Collison's group, showed that pancreatic cancers have one of the highest iron signatures, and that transferrin receptor elevation is associated with poor overall survival. So with this as a backdrop, we went and began to look at the role of N2A4 in pancreatic cancer, with knowing that autophagy is important and iron metabolism is important. Perhaps ferritinophagy is the link between the two. So Naira Santana Codina, a talented postdoc in the lab at the time, now with her own lab in Denmark, began this project with multiple collaborations throughout the lab, as well as through the Hale Center at Dana-Farber, and first noticed that the level of N2A4 in pancreatic cancer cell lines was significantly elevated when you compared it to a pancreatic ductal epithelial cell line. Similarly, the mRNA level of N2A4 was elevated when you looked at tumors versus normal pancreas tissue. So this suggested a high level of N2A4 and that there might be a high level of flux. But what about its role or its dependency? For this, we first turned to the Broad cancer dependency map, looking across cancers, including in pancreatic cancer, and we're excited to see that about half the cell lines in CRISPR-Cas9 loss of function screens were scored as dependent. Now, when we looked at the transcript level data in terms of the correlations between N2A4 dependency and transcript levels, what we were excited to see is that the most significant correlation between N2A4 dependency was that of an inverse correlation to ferritin heavy chain expression. So this would mean, where we surmise that those cell lines with the highest ferritin heavy chain expression, those ones that have the most ferritin and the most iron stored in ferritin, are most dependent upon N2A4, presumably to get that iron out to use it for proliferation. We also wanted to ask, using this data, is there something special about this N2A4 selective autophagy process that we'd identified in pancreatic cancer autophagosomes, or is it all of the selective autophagy adapters are important that we identified? So we looked across the selective autophagy adapters, now just in pancreatic cancer cell lines, and it was really only N2A4 that was a selective dependency in a subset of pancreatic cancer cell lines, as opposed to these other selective autophagy adapters. So we wanted to validate these findings in our own lab. So taking multiple different pancreatic cancer cell lines and knocking out N2A4, we saw a decrease in proliferation in a multi-day proliferation assay, decrease in clonogenicity. As expected, when you knock down N2A4 and look at the iron levels, so now we're able to use an iron probe to look at the free iron levels. You can see with two different knockouts, you can see a decrease in the free iron level about to the level of iron starvation with deferoxamine. And to show on-target efficacy of this knockdown with regards to this being related to iron metabolism, we were able to rescue the growth at 7 days by adding back ferric ammonium citrate. Now, we noticed that not all pancreatic cancer cell lines behave the same way, and including the cell line 898T, which really had no effect of losing N2A4. And we thought a little bit about, obviously the cell culture conditions are highly artificial and don't represent the tumor microenvironment that we see in pancreatic cancer. So we surmised that, there may be lower levels of transferrin, which is one of the main components of FBS, in vivo. And so to test that, we actually lowered the FBS concentration, and here we actually saw a differential growth pattern of the N2A4 knockout clones compared to the control clone. But what's the actual reality when you look at this in vivo, where there's a better modeling of systemic iron homeostasis. So planting these same cell lines, we actually saw that the result was more similar to this 1% FBS condition, where these tumor cell lines grew at a slower rate than the control lines. So this was exciting for us, but really the gold standard for evaluating the role of a gene in, say, the initiation and progression of a tumor is using a genetically engineered mouse model of pancreatic cancer that was first championed by Dave Tuveson's lab, where you're basically programming oncogenic KRAS expression as well as p53 heterozygosity within the epithelial compartment of the pancreas. And so we've paired this now with our conditional N2A4 knockout allele to create a so-called KPCN mouse model. And what we were a little disappointed to see, but not too unexpected, is that these mice still form tumors. And in order to look at the N2A4 levels, given the lack of immunohistochemistry agents, we derived cell lines from these KPC and KPCN mice and showed that there was a loss of N2A4, and there's a concomitant increase in ferritin levels and loss of this lower band that's indicative of lysosomal processing. In addition, there was an increase in the ferritin heavy chain staining, both at the pre-invasive and invasive stages, and an increase in the total non-heme iron levels. So this is both the free iron as well as the ferritin-bound iron. So what this suggested to us is that the N2A4-mediated ferritinophage flux is active in these in vivo settings in a murine pancreatic cancer mouse model. But what about sort of the initiation and progression question? So the way that we work with this mouse model is we actually monitor for tumor development using high-resolution ultrasound. And what we're interested to see is that in the KPCN mice, there was a delayed initiation of tumor formation in the KPCN model versus KPC. And when we looked at these mice over a long period of time in terms of overall survival, there was a significant improvement in survival in the KPCN model versus the KPC model. Some of this we think is related to the delay in initiation, but we also noted at the endpoint even that there was a decrease in Ki-67 as a measure of proliferative index, so it's likely that loss of N2A4 also slows tumor progression. So we wanted to ask then the opposite question. What happens if you elevate the level of N2A4? Does that alter initiation or progression of pancreatic tumors in vivo? So for this, we developed a conditional overexpression model whereby we're able to control the overexpression of N2A4 with a lock-stop-locks N2A4 knocked into the ROSA26 allele. Here we've overexpressed N2A4 systemically crossing with the CMV-CRE model, and you can see an increase in the N2A4 level and a concomitant decrease in ferritin heavy chain, both by immunoblot as well as immunohistochemistry in the liver. And remarkably, even despite turnover of ferritin and presumably higher levels of free iron, these mice appeared normal, had normal body weight, no real sort of discernible pathologic features, and had 100% survival at 9 months. When you paired this model, though, with the KPC mouse model, we noted that the expected frequency of offspring was significantly lower in this KPC N2A4 expression model. And when we did actually get a mouse, it was significantly lower body weight as measured at 3 weeks of age compared to the control genotypes. And when you looked at the overall survival, it was the opposite of the knockout condition. In fact, there was a significant decrease in survival in the KPC N2A4 expressing mice. All of these mice had to be euthanized by 3 weeks of age. When we pulled out the pancreata of these mice, we noticed that there was an increase in the pancreas body weight ratio. And in collaboration with Jonathan Nowak, evaluating the histology of these pancreata, we saw in the control mice, as expected, really no pathology or mild formation of pre-invasive lesions at the KPC mouse model at this 3 weeks of age stage, whereas in the KPC N2A4 expression model, there was a complete replacement of the pancreata with pancreatic adenocarcinoma. So it suggests that N2A4 was driving initiation and tumorigenesis even at this early age, basically 10, 15 weeks before we would normally see a tumor. We decided to then cross the N2A4 overexpression allele with a less aggressive form of this KPC mouse model without the p53 heterozygous loss, where normally tumors are only developing by, say, 40 or 50 weeks of age. But even, again, at the 3-week age time point, this pancreata were almost completely replaced with adenocarcinoma or pre-invasive lesions. So what do we think is going on here? We think that there's a critical interaction between oncogenic KRAS and free iron levels. And this is in part validated by another lab that had been interested in what happens when you have iron overload via a high iron diet in this KC-PDAC model, they saw a significant acceleration of tumorigenesis, and they had a complicated mechanism whereby they thought that there was ferroptosis induction in the tumor cell compartment that was leading to some paracrine signaling of macrophage compartment. Suffice it to say that they thought that there was a link to ferroptosis. So, remains to be seen in our mouse model, but there are hints that there's an increase in lipid peroxidation in our model by staining with 4H&E, but remains to be tested in a functional fashion with liprextatin, which is an inhibitor of lipid peroxidation that one could use in vivo in order to test whether or not ferroptosis is related to this initiation of tumors in this model. So, with these two mouse models, we think it established that there's some importance of N2A4 in pancreatic cancer, but we wanted to move on to understand, what is the sort of importance of this pathway with regards to, pancreatic cancer pathogenesis. And so for this, we went back to the Broad Cancer Dependency Map and looked at what are the top dependencies correlated to N2A4 dependency. And we were excited to see, as expected, there were links to autophagy, including some of the subsequently discovered upstream regulators of N2A4, TAX1-BPP1 and RB1-CC1. And then the rest of the codependencies were all related to iron metabolism in some way. First, transferrin receptor endocytosis, bringing iron inside the cell from outside, intracellular iron homeostasis, including genes involved in mitochondrial iron import, and then the use of iron in iron-sulfur cluster synthesis, as well as downstream of that, iron-sulfur cluster containing proteins involved in DNA replication and repair, as well as in a pentose phosphate pathway. To cross-correlate that, using one of our favorite techniques, we took a N2A4-dependent pancreatic cancer cell line, knocked out N2A4, and then measured what are the changes in the proteome as a window to understand, what are the proteomic adaptations to loss of N2A4. So in this volcano plot, things on the right are upregulated in response to N2A4 loss, whereas things on the left are downregulated in response to N2A4. What we saw, the vast majority of the proteins that were downregulated were all iron-sulfur cluster-containing proteins. So it suggested to us that N2A4 is critical in this N2A4-dependent pancreatic cancer cell line for maintaining the level of iron-sulfur cluster proteins. When we looked on a systems level using a GSA enrichment map technique, we were able to see that in blue, the downregulated pathways were related to mitochondrial metabolism, DNA damage repair, whereas the upregulated pathways were related in part due to iron uptake, which suggests a potential compensatory mechanism. We validated these findings on an individual protein level, showing that with loss of N2A4, you decrease the level of several iron-sulfur cluster-containing proteins, and you can rescue these levels by adding back ferric ammonium citrate. With regards to the activity of these iron-sulfur cluster-containing processes, there was a significant decrease in the activity of multiple different processes, ferroketolase, aconitase activity, and maximal oxygen consumption rate, along the level of iron chelation with DFO. How does this relate to in vivo? So we took a bit of a leap and took bulk tumors from the endpoint of the KPCN versus KPC experiment, and again evaluated using mass spectrometry-based quantitative proteomics in order to understand on a systems level what was changing with loss of N2A4. And again, we saw the same signature: decrease in mitochondrial metabolism, decrease in DNA damage repair, an increase in iron uptake. And on a select basis, there were several iron-sulfur cluster proteins we were able to see decreased levels by immunohistochemistry. So we really think that the critical function of N2A4 in pancreatic cancer is to maintain iron for synthesis of iron-sulfur cluster proteins. We haven't nailed down exactly which pathway that is using iron-sulfur cluster proteins is most critical, but we do think it is most likely related to aspects of mitochondrial metabolism, including oxidative phosphorylation. So all the experiments that I've shown to date in vivo have been sort of asking a question about initiation and progression, as opposed to what happens if you lose N2A4 in an established tumor. So there are no inhibitors of N2A4, but we're able to use inducible hairpins and show that in an established tumor, when you induce knockdown of N2A4, you're able to delay tumor progression. Now, as you've seen here, as well as in the genetically engineered mouse model, this is not a cure-all for pancreatic cancer. There are likely compensatory mechanisms that are ongoing here at the end of this tumor growth experiment. We looked back at our proteomic data to try and understand what might be some of these compensatory mechanisms. We saw, as expected, an increase in transferrin receptor, We looked in 2D culture systems whether or not you can add back transferrin with iron to rescue cell growth, and indeed there was a partial rescue. So that might be part of the compensatory mechanism, the endpoint of these tumors. In addition, there might be a decrease in the ferroportin level on tumor cells, so this would decrease iron export, so as a way of sort of rescuing the amount of iron in the cell. In addition, there might be non-autonomous mechanisms of iron sort of feeding to these tumor cells. We saw an increase in CD68 staining, which could suggest that there's an increase in iron-releasing macrophages. And so future work is trying to understand, on a tumor-wide level, what are the mechanisms of iron metabolism, both on a cell-autonomous and non-autonomous level. What about in humans? So the best that we're able to do is look at a ferritinophagy signature and show that the critical components of the ferritinophagy, N2A4, ferritin heavy chain, taxol BPP1, and RB1CC1, a high level of those mRNAs portends a worse overall survival that was validated in 2 different data sets. And so we do think that probably one of the important mechanisms of autophagy in pancreatic cancer, at least on a cell-autonomous level, is maintenance of iron metabolism via ferritin degradation. As I'd mentioned previously, there had been this observation that upon KRAS MAP kinase pathway inhibition, there was an elevation of autophagy. And so we were interested, we had a data set that we'd collected and published looking at what happens to the proteome upon KRAS G12C inhibition over time. And what we noticed, as others published, is that there was an elevation in lysosomal signatures, and we were excited to see that there was actually a change in the N2A4 and ferritin levels. So suggesting that there was some alteration in ferritinophage flux, and that, along with sort of global elevated autophagy, maybe ferritinophagy was elevated. Around that time, one of our collaborators, Rishika Pereira at UCSF, actually contacted us and said, we have this observation that when we look in the lysosomal proteome of MEK inhibitor-treated pancreatic cancer cells, some of the top enriched proteins in those MEK inhibitor-treated cells were ferritin heavy chain, light chain, and N2A4. And in a companion study that we collaborated on, they showed that one of the main aspects of this elevated autophagy in response to KRAS MAP kinase pathway inhibition is an upregulation of ferritinophagy. In this situation, what they posited is that this upregulation is critical for maintaining iron, again, for iron sulfur cluster proteins, but primarily for those iron sulfur clusters that are involved in mitochondrial metabolism, especially oxidative phosphorylation. And when they did the sort of synergy experiment with MEK inhibition plus knocking down N2A4, they saw this nice synergy. So at this point, we were excited that something that we discovered in pancreatic cancer autophagosomes, we've been able to show in mouse models of pancreatic cancer as well as in human data. It seemed to be that there is this extra reliance on N2A4, suggesting that this might be something that we'd want to target, perhaps in combination with KRAS inhibition. If you're thinking about targeting this pathway, we need to know a little bit more about the interaction. And so early on, we had nailed down where on N2A4 it interacts with ferritin heavy chain by doing some domain mapping. Suffice it to say, we were able to narrow it down to a small region in the C-terminal domain, and we're able to make alanine scanning mutagenesis to identify an isoleucine 49 and tryptophan 497 as critical for the binding to ferritin. We were able to reconstitute N2A4 knockout cell lines with either a wild type N2A4 or a double mutant N2A4 and look at the interaction with ferritin heavy chain. You can see this double mutant no longer binds to ferritin heavy chain. Looking at the localization of ferritin in your N2A4 knockout cells, you can see that ferritin is sort of widely distributed in the cytoplasm. When you add back N2A4 wild type, you can see the punctate appearance, lysosomal or autophagic localized, whereas in the double mutant, you're no longer able to localize ferritin to these punctate structures. On the ferritin side of things, it was known that N2A4 interacted directly with ferritin heavy chain, but not light chain. We validated that in a recombinant expressed system with N2A4 GST and looking at interaction with ferritin heavy chain, but not light chain. So what this meant to us is that looking on the surface of ferritin heavy chain, there must be something conserved on ferritin heavy chain, but not light chain, that is responsible for the interaction with N2A4. So we went and made alanine mutations across all of these conserved residues, and it had found arginine 23 as critical for the binding to N2A4. Fast forward, now So we also confirmed this in ferritin heavy chain knockout cell lines, where this arginine 23A point mutant was able to decrease localization of ferritin heavy chain in these punctate structures. So fast forward a few years, and we were lucky to collaborate with the Frank Lab now in this work that's in revision to understand what are the structural determinants of N2A4 binding the ferritin heavy chain. So we took the fragment of N2A4 that we knew tightly bound to ferritin heavy chain and placed them on cryo-EM grids at a 1-to-1 subunit ratio and collected single particle cryo-EM maps. We were able to determine the 3D electron microscopy map at a 2.9 angstrom resolution. Here we're zooming in on one subunit of ferritin heavy chain in brown, and we're able to resolve 16 amino acids of N2A4 in the orange color. And this was as expected given our prior biochemistry that we were only going to find a small portion of N2A4, and indeed the rest of N2A4 we weren't able to resolve, which is as expected given that there's many intrinsically disordered regions in this area. When we look at the within the ferritin nanocage, you can see that orange is the N2A4 binding domain on individual ferritin heavy chain subunits. And you can see that it binds on the long axis of ferritin heavy chain in this cleft, and that this cleft is at a distance from the 3-fold and 4-fold symmetry axes and at a distance from the iron entry pore, so suggesting that N2A4 is not going to interfere with entry of iron into ferritin. When we zoom in on the N2A4 binding site on ferritin and remove the N2A4 and look at the hydrophobicity of this pocket, in light blue are the hydrophilic areas, in orange are the hydrophobic areas. You can see that this is a mainly hydrophobic pocket. And again, sort of zooming in on where N2A4 binds on ferritin heavy chain, you can see that there's this hydrophobic area, especially centered around the arginine 23 residue. Zooming in, you can see the isoleucine 489 hydrophobic side chain interacts with the hydrophobic portion of the arginine 23 subunit side chain, and the tryptophan 497 of N2A4 is on the opposite end. We designed mutations that would further inform the function of this, or the functional significance of this pocket. A19Y, which would block entry of the N2A4 peptide into the binding cleft, asparagine N22 to alanine, which would reduce this hydrogen bonding, and then more conservative mutations of this arginine 23 in order to sort of parse out how important this hydrophobic side chain was. Tweenian in the lab generated ferritin heavy chain knockout lines, re-expressed N2A4, as well as the wild type and mutant versions of ferritin heavy chain, and confirmed that these mutants of A19Y and 22A and arginine 23A abrogated interaction, whereas the more conservative mutations, isoleucine and leucine, maintained some of the interaction. We'd also tested these phosphomimetic mutations of serine 114 in order to determine whether or not there was any alteration in binding, given that serine 114 had been shown to be phosphorylated in proteomic data sets. In addition, she looked at the co-localization of these mutants in cells, showing that the wild type co-localizes and these more ablative mutations decrease co-localization, where these more conservative mutations had some maintenance of co-localization. So with this information, we now have what we believe is the data set that we would need in order to actually target this N2A4 ferritin interaction if we were interested in generating an inhibitor of this process. So ongoing work in the lab is trying to understand the N2A4 ferritin interaction and whether or not we can disrupt this, how we can actually on a translational level target autophagy in pancreatic cancer, especially in combination with these newly discovered KRAS inhibitors, and also work ongoing is looking at new autophagy inhibitors that may be more promising than hydroxychloroquine. Just in the last couple minutes here, I'll just show one ongoing study where we're trying to understand the role of autophagy in the lysosome during pancreatic cancer tumor initiation, progression, and metastasis in collaboration with the Pereira Lab. So here we're using the next generation of lysosomal proteomic techniques. So here there's a lysosomal localized protein TMEM192 that you're able to affinity capture from cells in order to look at what are the lysosome contents. And the reason why we want to do this is that we know that the 2D cell culture system that we use is not really reflective of what's going on metabolism-wise in vivo. particular, it is not able to understand how the lysosome proteome evolves over the stages of pancreatic cancer initiation and progression. And in addition, we're unable to understand, with those 2D cell culture data sets, how the different microenvironments of the pancreatic primary versus liver or lung metastatic sites might alter the lysosome proteome and thereby tell us something about what autophagy might be important for at these different sites of disease. And so the Pereira lab has now knocked in the TMEM192 construct into the ROSA 26 allele, and we're able to cross this with the KPC mouse model. And you're able to see now this lysotag in red co-localized with the epithelial compartment in a pancreatic tumor and excluded from the fibroblast compartment. And so these mice are now breeding, and so we're excited to be able to monitor the proteome at early stage and late stage of pancreatic cancer to understand how the lysosome proteome adapts over time with pancreatic cancer. In work that's ongoing, in collaboration with the Pereira lab led by Thuy Nguyen in my lab, we took one of those KPCL, so lysotag containing pancreatic cancer cell lines, and are able to inject this either into the pancreas or generate liver metastases or lung metastases, isolate these tumors from these different locations, and then ask the question, what are the differences in the lysosome proteomes in these different locations that might tell us something about what autophagy is important for in these different sites. So we're always comparing these because we know that there's going to be a significant background contamination given that we're isolating these from tissues. We're comparing these samples from the lysotag positive cell line to a lysotag negative cell line. And here we're just looking at the input to the immunoprecipitation to make sure that there's no effect of the lysotag on the lysosome proteome on a global level. And you can see in the colored dots, this is looking at the abundance of these lysosomal proteins in the lysotag positive or the lysotag minus input. And you can basically see that there's no real change in the lysosome proteome. So that's basically a control experiment. When you then do the immunoprecipitation, as expected in the lysotag positive cells on the right here in the volcano plot, the colored dots are the lysosome annotated proteins that we already knew were lysosome localized. You can see that there's a nice enrichment in these different sites of the lyso-IPs. So we've now gone and done a multi-filtering step to understand, what are the basically specifically enriched lysosomal proteins from these different sites, compared across them to see what's in common as well as what's specific to different sites, and have zeroed in on, first looking at what are the sort of known resident lysosome proteins. So we already know these are lysosome proteins as a window into what might be the function of the lysosomes in these different sites. We've narrowed in on, what are the liver-specific lysosome proteome, in part because, liver metastases portend a terrible prognosis in pancreatic cancer, and so trying to potentially target lysosomal adaptation in liver metastatic sites might be a nice therapeutic avenue. And so as a proof of concept, we picked 3 of the enriched proteins in our liver proteomic data sets and have shown that in comparison to a pancreatic cancer lyso-IP, that there's significant enrichment of these proteins. So work ongoing is to basically then validate what might be the functional significance of these liver-enriched lysosomal proteomes. So thank you all for your attention. Just the summary, we're starting from basically the initial question of what are inside pancreatic cancer autophagosomes. We've discovered a new selective autophagy pathway that's critical for maintenance of cellular iron homeostasis. It's also a major component of the tumor cell autonomous dependency on autophagy in pancreatic cancer. And so this leads to potential new strategies for inhibiting autophagy, either alone or in combination. And so that's work ongoing in the lab. There's many people in the lab that have contributed to this work. The pancreatic cancer work was led by Naira Centeno-Codina, but with a whole host of collaborators within the lab, as well as here at Dana-Farber, including the Aguirre lab, Jonathan Nowak, as well as labs outside of Dana-Farber. Thank you all. Happy to take any questions.